This is a Zoom from the first time I tried to record this out of 3 times. And the sound didn't come out, but I did want to show this activity because on the day I recorded live, that when the sound did work, I didn't get to do some of the stuff I often do. So anyway, you can see this is my board. All my students are on here. You can see their names. So there's 8 of them. 4 of them are following me around. And I've turned on the thing where I can see them. So I'm creating some boards and I'm asking them basically, and I'd be explaining this in class too, to get, turn to partners. Yep, here we go. And then they're doing a thing where they're coming up with characteristics that animals either have uniquely or they share. And so if I want to, I can follow and see what the students are doing on the board. And so they're working together and you can actually turn on the cursor so you can really see where they're working. And so they're just putting little stickies on here in order to come up with some ideas. You can see that they've come up with more ideas and they're working together, and then we can use those to talk about stuff. This is like brainstorming essentially with partners. I can put little tags on them, thumbs up, or classifying them as different things. And so then we can keep going back and forth while we're writing the notes, going back to their proposed answers. Then I also had them do this thing. So it's essentially a table, but it's made out of note cards and or sticky notes on the board. And so these are different, like each category, and there's different characteristics. And so I have the students get in little groups, and then they're going to take stickies and fill in the different parts. So essentially making a table, but with sticky notes. And that way everyone can eventually see each other. So they don't have to take photographs of it, and pretend they're going to look at their photos again on their phone because that's unlikely. Instead, they have it contained on the board, and then I can project it for everybody. Everyone can see this on their computer as well. And essentially, and this is me just messing around with it, but essentially in their little groups, I assigned each of them one of the groups, and we've already gone over all those in class already. I can see the students working over there. Oh, and I also have a timer on that they can see, and then I have music on that they can hear as well. It just didn't record the sound. You can see everyone working. And the sound started, and I don't know why it started now, but you can see the students have filled it out, so they worked with partners. Then the sound started picking up. In any case, I recorded a different day, so you had the whole thing with sound. But this is one way we can use the board in order to make information like this, and then we can go through and I can modify stuff. We can to correct it or to put more detail on there, and then eventually they have all this stuff on their board, and it's searchable and all that kind of stuff. It's also I have like a table of contents where they have all the previous board or all the boards on there, so they can go to there, and that's what I link to on D2L, so they can get to all their different boards from that spot. And then what I also wanted to show, we're doing these groups. We're doing figure making for an experiment we're doing for the whole semester. So essentially I've created a series of build-up figures, and they work together to create these. So they didn't have all these lines to begin with. They had to make all the lines between the groups, and I give them information. Eventually they're going to put them on a little model, the cards, and see what they expect. And then I can go and look at each group. So this is like Josh and Isabella, and you can see they're on the board. I can watch what they're doing while they're doing it. Of course, I can go talk to them as well, but I can watch. And then they'll tell me, hey, Professor Scher, can you check my stuff? And so then I can just pull up theirs. You can actually see that I have everybody's up here. So I can just pop between people, and that way I can help them, or I can make sure they're on track, or I can give comments. So I'll put comments on the board after I review them, asking them, hey, take a look at this spot, make sure you pay attention here. There, I was giving a comment there. And this is all very kind of intuitive on the board, really, because all the arrows snap. There was a comment with me. There it is, with my face on it. So if they click on that, they'll see the comment, and then they can adjust things as needed. Anyway, so it's just a way we can, this is another way I use my boards. And so this is something for lab we're doing there. Just to introduce the class that was recorded in its entirety and give you some background on that. First of all, the room is quite large, and it has incubators and fridges that are loud in it. So I had to talk quite loudly. And then the microphones are kind of different places in the room that are picking up the sound recording. I think when you're on a Zoom call, it's fine because we tested it. But with the recording software, sometimes, or the Zoom recording, sometimes it modulates stuff out. So you'll hear me just fine. And I've tried to edit it. I didn't edit any content, but I did put the timestamps in and I did mash up the videos, and I did try to modulate the volume there. You can hear the students sometimes you can't, but I usually repeat what they said. And if you just like stick with it, then usually you'll hear the students. And it depends how many people are talking to, because sometimes when there's more people talking, it just like doesn't record anybody. So I think that the sound is not the best thing in the history of the universe, but it is doable, and I think you will understand how the class went. My class is 2 hours and 50 minutes. And obviously that's a lot. So I've made a table of contents that like highlights some points of different kinds of things I do during the class. So there is a decent amount of lecture, but I do intersperse with questions for that. I also do have a little activity that they're doing. And then we do dissections of an earthworm at the end. Then I have students who stay for like 20 minutes after class and chat, which is all very interesting and good examples. Even the lab, I know people, when they observe, they don't stay for lab, but I do think it's quite important. It surely shows you how you interact with students when it's not just like a formal, I stand and do my thing, or you're doing your activity and I'm coming around with a specific, answer set. It's very organic and fluid, and I just think that it's an important thing to watch as well. So take advantage of the timestamps in the menu, and that way you can jump around if you want, or you can always play it on faster speed, or you can just like cook dinner and listen to my 2 hour and 50-minute class and see what you think. Thanks. All right, so today, thank you for chilling for a little bit. Today we're going to finish the animal characteristic development, that kind of thing, and then we will do start getting into the animal groups. We're going to do the dissections. I have different animals on the desks here. Like different collections, which eventually, I don't know if we'll get to today or not, but eventually we'll sort of see if we can classify them based off the characteristics we talk about in class, like the major identifiers. I will say for this exam, for exam 4, we're not doing, not too bad. All right, I'm not doing too much A&P, anatomy, physiology, or structural function. We'll talk about the parts that I want you to know for the different groups, but it's not going to be as extensive maybe as you might think. Like here's we always have our learning modules on the board, as well as our notes to download, and then papers and supplemental reading and stuff. So here's our objectives, and they include some A&P stuff. So mostly I'm going to cross out a few of those, I think, and we'll do some of them, and then we'll talk about the other stuff in combination with animals 2, which is the deuterostomia groups. All these are non-deuterostomia, so we'll just kind of overlap those. It's not a whole lot here, but like excretory organs or the waste, we're going to save that. So just FYI, a lot of what we want to know for the exam is really like, how do you identify the groups? So I'm going to give you like pictures of the groups, things that you've never seen, and you're going to match them with, what do you think this is? How are you going to do that if you've never seen it? You're going to look for the characteristics. Know something has certain characteristics, you'll be able to match. I'll ask you questions about, here's the defining features in words of the groups, and how does that, then you'll be able to match those to the groups, whether it's the clades or the phyla or the classes, whatever kind of level we're on. Yeah, and we're going to put those together in a different board, the diversity board, in a table. So you'll be able to study that table with the most important points there. But first, we need to finish up our animal characteristics, animal diversity, not diversity yet, but the animal characteristics that we're looking at. We've done our tables already and gone through, I believe, all this stuff. Let's see where we are. Okay, yes. Okay, so we are getting to, we did symmetry, and now we need to do development. So last time we talked about the different body structures, basic ones. We've got asymmetry / non-symmetrical or variable ones. We've got bilateral symmetry and radial symmetry. And so let's see what those go along with for development. So I'm going to, let me see. We'll do our coloring stuff. So you can get out those sheets we had the other day. If you weren't here, I believe everyone was here. And now hopefully some more people will come in. But They're on the side there. When you pick up your colors, you can do that as well. This is just a little more efficient than me drawing everything because it takes a little bit. I can draw it, but it takes a little bit of time to do that. So I will draw the life cycle, but I think it's a little easier to color these things. I do have them on the board if you prefer to use them from there. If you'd like to do your stuff digitally, you can download that from the board. They're on the top left. And of course, if you get to the menu, you can just push the download button and they're in PDF format. You can always save them as TIFF if you need to. I'm gonna do them on the board today instead of in the other program. I mean, I know sometimes it's obvious, but the presentation feature of this is relatively new. So, of course, I can always just zoom in on the slides and draw stuff and we'll talk about them that way. Or you've noticed, right, that I do the present and then you can follow it if you're on your computer. I'm just curious from your perspective, is there a preference of which one? And of course, when you're doing the presentation on your computer, you can always click off, right, to get out of it, to see it, or just not opt in. Does anything feel more or less efficient? Or
I follow along so that I can make notes, maybe you're going a little faster than me, so I can get behind you with it.
yeah, does it help to be on the so it would help to be not on the presentation because that's me advancing it. I suppose you could go, or
I join us later.
okay, keep where we're at. Anybody else have thoughts about that? Equal? No preference? Let me know if you think of something. I think I'll do it like this. All right. I'll do it in the presentation. And I'll bring in some videos later to show you of these development stages. And we'll be able to see them, but right now I'm just going to talk about them in a static way, I think. So, we talked about life cycles and we'll draw the life cycle in a moment, but in a cycle form. This is just a linear form. And animals are more familiar to you, hopefully, because you talked about them with 1406 and such. And you live your life as an animal, so we kind of know this. So if we're starting with the zygote, which remember is the fertilized egg, we talked about zygotes in the context of fungi, we talked about them in the context of plants, so we're pretty familiar with that. And then we have the processes, but we're talking in more detail for animals about the actual development stages. It's not like there's not stages in plants and fungi, in plants in large part, but we didn't talk about those cleavage stages and stuff, but we're going to do that for the animal groups and learn what these different parts mean. Okay, so I will do this in a cycle since we usually show it that way. But to preview it, we're going from the single cell zygote. All right, so the zygote, do we remember what ploidy all the zygotes are? This is the same as we talked about before.
Haploid.
Okay, and what does a zygote form by? What process? How do you get a zygote? Get it from fertilization. So if you have fertilization, what does that mean? Combining cells from or parts from two parents, right? So if you were to think about that, how many copies of every chromosome would you have if you had two parents that combined the chromosomes in the zygote? Two copies, because we're getting one from, in this case, the egg, one from the sperm. So if you have two copies, what do we call that?
Diploid.
Diploid, because of the di part, right? That tells us. So zygotes are diploid because they're from a combination, in this case, of the egg and sperm. So we have a one cell diploid zygote. We have, and that's the same as we've been talking about. So that's here, cleavage, which just means division. In other words, mitosis, which I've written up here. To make this thing called a blastula, which is like a ball of cells, that more and more division to make this ball of cells. And I'll draw this in a minute. Then we get that ball of cells and it starts folding inward because it's like you have a ball, now you need to make the body parts. I have models of them over here. There. But it's going to start folding inward and make eventually a gut, whether it's a gut with a full gut with 2 openings, the anus and the mouth, or sometimes it's just like a pouch with just 1 opening. And then that's going to end up folding and rearranging more in order to actually form the organs. And then that'll actually grow into an embryo, and then that'll grow into an adult eventually. So we're going to talk about this process a little bit. You can see the different stages that are happening there. So this you grab a couple of those. I want to block myself too early. Thank you. I have it. Okay, do it. Perfect. So if we start at the top, and I'll do it in the cycle form, as we just said, we've got a single cell that is called the zygote cell, and I'll do it in our multiple colors so that we can see the processes in a different color than the like the structures. Zygote, we know that that's 1 cell, and we know that that is diploid, so we'll do 2N there. Then that's going to divide. Actually, make all this the same color. Blue. All right, because I want to use purple otherwise. That'll divide, and first there's obviously 1 cell goes to 2, 2 goes to 4, 4 goes to 8, and so that's what you're seeing here, right? This is the zygote, 1 cell here. That's going to divide. Which way is the? Yeah, this way for you. Into this is now 2 cells, and then that will divide into 4 cells, and of course that will divide into 8 cells. And then at this point, you have different. Even at these earlier stages, you can have different sort of arrangements of how they divide, but we'll see that in a minute. And then you have 8 cells that'll divide into 16 cells, and then that'll divide more until we get to this next stage, which is a ball of cells, which is represented both here and then also a cut through in this case. So that's the same thing here. That's the blastula. That's the next stage. So we, instead of drawing out all the cell divisions, I'm just going to draw it to the blastula. Well, let's draw it over here. I'm going to do another one. So you might want to draw this to the side or kind of below it. All right, let's do it here. Then we'll draw it in the little stages. Okay, zygote. Then eventually we get to the blastula, which I'm going to represent by like a circle, and I'm just going to put 2 circles and lines. Each of those lines is a cell. So I'm going to put a dot in there for the nuclei, so you can hopefully tell that that's just, this is a cut through or a section through the middle. If you were actually to look at it whole, it would be like a sphere. This is actually, once it gets to the end, the blastula is where you've heard of maybe embryonic stem cells, ones you can take out and then turn into anything to like help people who are missing, have damage, so that you can like make new cells from the embryonic stem cells that would allow them to replace those cells. That's when they're here. So embryonic stem cells will be when it's at this stage. It's a little clump of cells inside in humans, and that's what gets taken out there. Okay, so this is a ball of cells at this stage. We call that, though, a blastula. So we'll write that word here. Whenever we have the word blast, it's usually referring to early development, like early embryo stages. So blast is referring to like germline, which is the reproductive stuff. So this is many cells, and it's still 2N. pretty much most of the animal life cycle is going to be diploid. So the question becomes, if we go from one cell to many cells, 2N to 2N, what kind of division would that be? We're going the same ploidy. We have mitosis, yep. Make sure you speak up so that the recording captures your voice too. It's not just you. But yes, you're right. Mitosis, which we call cleavage here. That's just the special name for it in the case of animal development, but it is mitosis. So we call that the cleavage stage, all those stages where it's dividing from the one up to the ball of cells.
Stages where they're called cleavage is a dividing stage.
Yes, pretty much all that happened. Well, that's not entirely true. So gene expression is changing during cleavage. So stuff's becoming different from each other, but the majority of what's happening during cleavage is the mitosis, to go from one cell to many cells, hundreds of cells. But just in this ball. And then you're going to get that ball. Now you need to take that ball and start making it have certain shapes, right? Because an organism, those shapes are eventually going to turn into the organs. So this is when we get to that gastrula stage. So I'm going to draw it like this because it's folding inward. This is a cut through again. So if we looked at this in 3 dimensions, it's, I don't have one. It would be like this ball, and it would be like the bottom pokes in, or it so pokes in, kind of like if you took a balloon and stuck your finger in the balloon, not through it, but just through the balloon. You can imagine that's pushing like kind of a cup structure up that elongates into like a long pouch there. So it basically starts folding under and folding inward to make this inside structure there. And like I said, I can put up some videos so you can see the different stages. It's pretty cool. This is the stuff I used to do for research, right? Is make pretty videos on this stuff. So I'm going to do not actually 2 layers. This is just going to show that it's made of a lot of cells. So I'll just put a nucleus in each one. So hopefully we realize that means they're all cells. Okay. So this would be called the gastrula. Gastro. Are we familiar with GI doctors? You know, like gastrointestinal, that's what the GI stands for. Gastric bypass. Can you think of like what that gastric was referring to generally when we use those words? Stomach stuff, gut stuff, right? So in gastrula is the gastrulation, which is what's going on here. That's the process that you're starting to form the gut, which means a variety of structures depending on the type of organism. So it could be stomach, esophagus, intestine. It could be crop, gizzard, stomach, or proventriculus. It could be a lot of different things depending on the organism or the type of animal, but basically that's what's starting to form here, this tube. So this process is gastrulation, the formation of the gut. And that happens by folding of the tissues, the different, the whole ball. You're rearranging cells. And rearranging cells so that you get the tissue rearranging. So you've got folding. And then we have differentiation of those cells. So up to the blastula, cells are pretty much, well, they have some differences, but they're kind of uniform at that point, which is why you can use those embryonic stem cells because they're not like differentiated yet. But once they get to the gastrulation stages, then they're starting to differentiate. So if we wanted to think about differentiation, we could define that as like specialization. The cells start getting specialized.
This is across the board for most animals.
Yes. Except for sponges.
I noticed that sponges.
I think they don't have a gut in the end, but they do have to do cell shape changes. They do have multiple cell types. So even sponges would do differentiation of their tissue, of their different cell types. And that's how you get, in that case, not tissues, but you do get the different cell types doing different things. Most animals, the differentiation is going to result in different tissue types. So if we were to think about this like in 3 dimensions, it would be something more like this. Like a hole here, and then inside you've got this tube forming. Push-in tube there. Like dots mean it's inside. A little cleaner. And that is like this. Still cut through, but you can see, right, that there's like a, it's not a hole, but a part here, it's pushing inward, and this is pushing up. These are just cells, of course, pushing upward, and eventually it'll get to this situation where it's pushed all the way up and made this this longer tube inside, and eventually in a lot of animals, this tube will connect to the other side and form a second opening. So you'll get mouth and anus, depending on the type of development, it could be either direction, but then that'll be the gut, which can then turn into different gut organs if there are specialization there. So that's like your beginning gastrula, later gastrula. So early, early gastrula, later gastrula. And I'll put these in order again on the side or in the back so that you can see them if you want to look at the different stages in those models. So then we're going to simplify the drawings here because draw the stages, but we're going to go all the way around to forming an adult. We're just going to, obviously goes through embryonic stages, sometimes the certain animals fetal stages, and then ends up being an adult. Now, does that mean like jumping to the adult right away? No, it's like, you know, the offspring when it's born. So a baby counts as an adult because it's the body form. Or it might count as a larvae. So we'll see those two options. Gastrolite, we should say it's many cells. And it's also diploid. Now, one option is because this or becomes an adult, and that process there, well, let's call it organogenesis. It's going to be the rest of development, but a big process in that is organogenesis, generating organs, right? So the tissue types that are differentiated now will form different shapes, get further differentiated, have different functions, and become the different organs. Genesis means like creation or formation. Adult, of course, is many cells. Diploid. Larvae or larva, singular, are many cells. Diploid. The larvas is not a word. Larvae with the AE at the end, LARVAE, that's plural. Larva is singular. Up a little bit. Do you guys know any examples of larvae in animals? Types of larvae? Say what? Beetle is going to have larvae. It depends on, usually the larva of a beetle we call something different than a beetle. You're right, it is a beetle. It's going to live in the ground. like a little wormy sort of thing. Might not know which ones. Grubs, yeah. So we often call them grubs. Have anyone seen grubs before digging in the ground? They're like little white, fat, like wormy things in the ground. So those are grubs, and they will turn into beetles. So that's the larval form. What's the larval form of the butterfly called? Caterpillar. And moth would also be the same. Larval form of what else? Flies. Maggots. Right, so larval form is the first stage, usually pretty wormy. They're not worms, but they look like that. So that's the first stage in some animals. So that's why it's an or. We don't do a larval stage, but some do. And then they will change from the larva into the adult. And I think most people are familiar with, caterpillar turning into a butterfly. What do we call that? Okay, that's pretty technical. What's that? This is true. So it does form pupa in different instar state. Well, the instars are the larvae, and then it develops inside of it. When it's in the pupa or the chrysalis or the cocoon, all those things are I guess the pupa is the thing, but it's inside. What's that process where it's like redoing all its tissues to make the butterfly? Metamorphosis. Have people heard this word before? Metamorphosis, and that's the larvae, like a butterfly undergoes metamorphosis, right? From the caterpillar to the butterfly with the wings. So that's the process here, taking the larva to the adult in some animals. And mostly it's going to be not only insects, some insects or many insects. We've also got this in like some of the echinoderms, like sea stars and brittle stars. We have this in some of the cnidarian, like the ones with the stinging cells and tentacles. So you see this across the animal kingdom. It's not just an insect thing. But it's common in there. And it's pretty interesting because I don't know, I didn't realize this for a long time, that all the body tissue breaks down, and then there's stem cell populations inside the larval body, and then they grow into a whole new organism. So it's not like the caterpillar like morphs its existing thing into a butterfly. It's like all the stuff that's the original caterpillar. It's like it just essentially dies, if you will, and then those stem cell populations inside of the caterpillar that were hanging out there, they get activated and then they turn it into the whole new thing.
And it's pretty crazy looking. I saw an example outside one of a Asian lady beetle. They're kind of larval state of bins. So it's like they're what used to be their body just like that.
Yes, and that's the same with butterflies too. If you see the chrysalis form, it's like the body turns into the chrysalis. It's not like that it builds one around it. I suppose that's what I always thought. Cocoons can be built, but I think this is what you're saying too, right? Is that the actual larva turns into the structure, which then basically ends up making the thing itself, the inside of it, makes the new adult version of the organism. I've never seen a beetle do it before. I've only seen butterflies. My parents raised like 600 monarchs one year. Yeah, it was wild. And so I saw a lot of those. I do have some videos. I'll try to pull some good ones. Because it was always like, you waited in the morning, because they like to hatch in the morning, the chrysalises. As you can imagine, it was pretty cool. But it's not only cool for Simon, who's biology. Like, people would come over the house and then they'd go and buy their own milkweed and they'd take some larvae and grow it because the wasps were getting them. So they were bringing them all inside. So those wasps like to take the caterpillars and eat them to their young. So in order to protect them, they started bringing them inside. They got this cage. It was pretty wild. I mean, they do it, but not 600. Then Harvey happened, and then it washed away all our plant and whatever. But it was an experience. And I just have to say, Isabella is an insect expert. So ask her any questions that you have. We talked about it before. She's going to bring us some specimens. Well, I'm spelling this wrong. Or maybe not expert if you don't want to be called that. Enthusiast, we could say. I mean, compared to the rest of it, the rest of us, I'd go with expert level. So then, and that's a big process here with new tissue differentiating, all the things. It doesn't go through gastrulation again, but it does organogenesis, totally makes all the new organs. Okay, so then you have adults. And we talked about the other day that not all animals do sexual reproduction, right? Some do asexual. We talked about the advantages and disadvantages. You guys made a nice table about that. Most animals will be able to, I'll remind you, or if you weren't here, yeah, this part about sexual, asexual reproduction. We made this nice table. This will be, a writing thing for you. Short essay on the exam. But most animals do sexual reproduction, which means there's two parents, or in hermaphrodites, one organism that makes eggs and sperm. If they can self-fertilize, then it's just one parent, but sometimes they still have to cross-fertilize with another organism. Usually, though, the sexes are separate in most animal groups. So we'll have adults, we'll have another adult, one that produces eggs, one that produces sperm, or two that produce both. But you'll end up, this is the sign for female. This is the sign for male. Or hermaphrodite is both together. So you could have options here. But in the end, you need an egg and you need a sperm. Maybe let me draw like this first. Because I want to get to the fact that we have a sperm cell and we have an egg cell. So I just mentioned, but how many cells is a sperm? Say again. One cell, yep. So one cell. And what ploidy is our sperm? We talked about this in plants when we talked about key concepts, right? It's a haploid. Yep, good. But that told you all those key concepts will just keep coming back, right? And that's the nice thing, because once you understand the key concept, as we'll see, then you know what process is happening. And the same with eggs, right? You might hear an egg called an ovum, plural ova. Suppose one N is going to be more is cleaner, but haploid is the case. I'll just do N. You guys know that by now, the abbreviation for haploid. Okay. So we end up having an egg and a sperm, whether that comes from two separate individuals or one individual. This adult is also 2N, right? So now the question becomes, using key concepts, what process does the adult do inside its body to make the gametes? As you know, eggs and sperm are called gametes, right?
Talk about meiosis.
I'm talking about meiosis. How did you know that?
That's like those things.
Yeah, half the ploidy, right? We know 2N to N is always meiosis. And probably familiar with this from Bio 1. But even if you forgot, you're like, okay, I know some division because I got to make new cells. What's happening with the ploidy? It's getting halved, right? So we know that that's meiosis, is the process there. So this is going to happen in testis tissue to make sperm, ovary tissue to make eggs. Those are called gametes. So that's happening inside the body of the adult. Get crazy? No, it's good. All right, so then An egg and sperm, sometimes people say they fuse or combine. Technically, the sperm ends up donating its nucleus to the egg. Oh, and I should mention in all animals, the sperm is flagellated. Remember in plants, we had some groups that had flagellated sperm and some groups that didn't, right? The bryophytes and the non-vascular, seedless vascular plants, right? Ferns and lycophytes, they all had flagellated sperm. Then we got to gymnosperms, angiosperms, which did not because they had the sperm inside the pollen. But in this case, all the sperm are swimming. And the eggs are not. The eggs are much larger than the sperm too, like a lot. If you had an egg this big, the sperm would be like that big. But I'm not drawing it to scale. Okay, so then we have those two essentially in the end combine their nuclei to get to the zygote. And I think you all know that process there, egg sperm making new cell. What's that? Fertilization, good. Which is, combining two haploid cells, which makes sense because we get back to a diploid cell, the zygote. It's also important. Let's write this down. And add something to the gametes in a minute. Gametes are, or since gametes are produced by meiosis most of the time, how would they compare to each other genetically? Like each sperm produced by an individual, and each egg produced by an individual. How would the genetics be when you compare all of them?
They would all be different.
Okay, why would they be different? Spindles pull, so the lining up, opposites like with homologs before spindles pull. And what's the other? There's like two main things. Yep, so that's independent assortment. What's the other part earlier in meiosis where they get mixed up too? Crossing over, prophase 1. Yep, so both those things are making mixes, which then, and of course, there's only half the chromosomes in each one. So that means you get different, you get one of each chromosome, but different ones potentially in each egg or sperm. So let's make a note of that. Whereas before, in all these cases, it's all genetically identical cells. Here, we're getting genetically varied. So all the sperm and all the eggs are different, even within a hermaphrodite. So that's the thing, right? Hermaphrodites are not, that's why I don't classify it as asexual reproduction. Because hermaphrodites, even though they produce their own eggs and own sperm, they're still all varied inside the body. So every egg it makes is different. I mean, this is the same with species with, two separate sexes, right? Like all the eggs you make, if you have ovary tissue, are all different from each other and sperm. So let's write that here. That's important. Genetically varied. And then we know too, the variation also comes from fertilization because it's like, random egg, random sperm, as long as they're healthy. And so that mixes up the genetics more. And we talked about all three of those generating variation in sexual reproduction. We talked about that in Unit 1. If you remember about that essay that was like, tell me about ways genetic variation is generated. And then one of them, you could say sexual reproduction, and then these kind of processes of meiosis and then fertilization. That's how you get new offspring. So essentially the zygote is the offspring. So that's the animal life cycle, which obviously, as per usual, will be on the exam. Any questions about that? Pretty more straightforward than like fungus life cycle, I would say, right? You say, oh, yes?
Yes.
Yes.
Indubitably, yes.
Indubitably. Oh, great word. Yeah, that's a great word. I do want to draw, I'm not going to draw all the stages of cleavage and gastrulation like I sometimes do, but I do want to talk about a few names of parts of the blastula, like a terminology, and parts of the gastrula. And we will draw 3 stages of the gastrula as well. So the blastula, which in some animal species they call the blastula the blastocyst. And like in humans, they call it the blastocyst. Cyst meaning, well, not really cell, but I don't know. It just sometimes it is. But if you say blastula, that covers everything. I just want to let you know if you take A&P or you know go into the medical field, blastocyst, it's blastula, just in only certain organisms. Okay, so this is the blastula. I'm going to draw this time a bunch of cells in a circle. As you remember, this is a cut through again. Or section, section cut down the middle. So we need to know some of the characteristics. So this inside, I'll color it in. It's called the blastocoel. C-O-E-L. So you pronounce this like seal, blastocoel. Seal is referring to a cavity, like an empty space, a fluid-filled empty space inside something. So blastocoel is the fluid-filled empty space inside the ball of cells that is the blastula. And let's note that I kind of mentioned this before, but the blastula is a hollow ball of cells. The inside is full of fluid, though. It's not like empty space, mostly water. So it's hundreds of cells by the time it gets to blastula. And at this point, it's relatively undifferentiated. We could say that they have minimal specialized jobs, specialized structure, specialized function, specialized. Say that they don't have an identity. They might have some identity. They would, but not like there's still a bunch of things it could turn into at this point. How would I say that? I don't say no identity because there is some limited identity. Does that make sense? Makes sense. Minimal, that's not right. Not much identity. I don't know how to say. I don't know. We'll do that if you think of something better. But yeah, I think you understand what that means, right? They're not like it's not a kidney cell yet. It's like a type of stem cell, essentially. At this point, it's a bunch of stem cells. Yeah. The blastocoel is this blastocoel. It's inside, fluid filled inside. And if anybody wants to take a picture or a series of these and throw it on the board, feel free to do that. That could be nice. Then we come to the gastrula, and this is when we'll do a little bit, we'll do our coloring. Yeah, we'll do our coloring next. Gastrula is the thing, right? That's what the embryo is called at this point. So if we look at an early gastrula, make it prettier this time. Let's see. Draw the circle first. I'm going to dip it down a little bit. Bring it over. So instead of being flat, because that's accurate, it's because it's rolling in. So it's going to be dipped down a little bit. And I'm going to draw those dotted lines in the inside again. And not very far up yet, because it's early gastrula. Sorry, that's too far. And on your figure to like label or identify or put in order, you know, your animal's life cycle, I'm going to have early and late gastrula. I'll have, you know, 2 or 3 different ones. So you need to put them in order. So let's look at parts here. We still have, what color did I use? Blue. All right, we still have the blastocele. That's all out here. So the blastocele from the blastula, it's getting pushed into, right? But we still have the blastocele. Then the tubey thing that's forming, this part, so blastocele is out here. Then here, once this is forming and it pushes up, that inside here, the little cup tube thing is the arc enteron. So it's like, you can think of like an arch. It's not said arch, it's called arc, but it is with the CH, so arc enteron. And tero means inside. So the arch inside the body. This inside here is the arc enteron. Tero is inside, said with a K sound. Which is going to be your future gut. Like I said, gut is actually a technical term. I know it sounds like a slang, but it's actually a technical term, and we call it gut because in different animals, the digestive system, the tube part of the digestive system is different things. So like us, it's your mouth, pharynx, esophagus, stomach, small intestine, large intestine, rectum, anus. But in other organisms, it's different parts there. But essentially, it's the gut is the tube part of the digestive system. And then this part where it's pushing in, like where your finger's going in the balloon, as I was talking about, that part is called let's see, blastopore, sorry. Pore meaning, hole essentially. And that's going to be one of the future gut openings. Whether it's the mouth or anus or gastrovascular opening, it depends on the animal. Usually, I'll say there are species let's finish writing this. There are species that form a different opening. So they'll do this process, then the blastopore will close and another opening will open up. Complicated, of course, like biology, there's always like a bunch of exceptions. We're just going to go with the generalized, most common thing. The blastopore will turn into the mouth or the anus, or if it's a not complete gut, it will turn into the gastrovascular cavity opening, like the one mouth-anus combo. Now I'm going to use some colors, and these colors are going to be consistent. And we're going to look at the differentiation stages. So that was sort of like gastrulation. Then later, during later gastrulation, so there's going to be a later gastrula, and we're going to show differentiation occurring. I'm going to use blue. I'm going to use orange. You can use yellow, whatever is easiest to see, but I'm going to use blue, orange or yellow, and like a red or pink. It's my magenta color. So this is consistent with your book. The book uses these for the different germ layers or embryonic layers that'll turn into different body parts. I can show you that here. So you'll see they're using blue, and they're using yellow in this case, and then they're using the red. So we're going to consistently use this so we match, because there are 3 tissue layers or 3 germ layers. Germ means like, you know, embryonic early development. So 3 embryonic layers that then are going to be certain types of things. Now they're differentiated to become different. And they even, for the organs that they can turn into, they even use those same colors. So blue is going to be, what do we want to draw first? Let's draw the inside. Let's draw the archenteron. So this is going to be your orange or yellow. So that is going to be that tube inside. I'm drawing it, obviously, cut through again. So we're going to call this inside, and I just mentioned this term. I'm going to do a key. Do you remember what term I just said was meaning, and you might know this, meant inside? Inside something? Endo. It's also endocytosis. You remember that from Bio 1, that taking in process, endo. Yep. So this is called the endoderm. Derm means skin. This is not, you know, in this case, it means covering. Okay. So layer, I guess we could think. So this is the inside layer. The inside, what we call a germ layer. So these germ layers are in the embryo. They are differentiated now. So there's only certain things they can turn into, pretty much. They're limited to what tissues they can turn into. Tissues, and later, obviously, organs. I guess really cells turn into tissues, so we'll do that. They can become. There are a lot of them. So each germ layer, there's just 2 or 3 in each alum. Each germ layer can turn into a number of different types of tissues. And let's actually add limited to which cell types. That's really the first step. Cell types, and then tissues, and then organs they can become. We're going to go like on the organ level pretty much. Okay, so endoderm inside, the inside skin layer, if you will, or covering layer. This is going to turn into the inside, the innermost tube of the body, which we call the gut. Can turn into a lot of other stuff as well. You can see that in the notes, at least in vertebrates, in the vertebrate group, where the endoderm can turn into. We're going to be worried about, and some of these are only in vertebrates, but we're going to be really thinking of the lining of the digestive tract. And then you can see that here, right? Here's the different embryos. You can see the archenteron forming in all of these. So that's early gastrula on the left. Early gastrula on the left, and then the middle you can see the archenteron forming, and then eventually it's going to become a full gut tube essentially, and then you can see here in the adult organisms, this is a gastrovascular situation at the top where it only has one opening, but you can see this is lining that gastrovascular cavity that came from endoderm. You can see the gut over here, that tube on the inside, that tube on the inside here, which comes from the endoderm, and then on the bottom, you can that's a planarian flatworm, platyhelminth, and then you can see here, there's your gut in a deuterostomy type development, and that's this case, I think it's a sea urchin. Yes, it's a sea urchin. So, but we're gonna just keep it simpler, I think. So we're gonna say endoderm to the gut. Okay. Then we're gonna let's use our blue. So blue is gonna dip down a little bit, come around, and then dip down on the other side. So it's connecting to the endoderm. That's sketchy. Hold on. And we talked about, you probably know two words or two parts of words that mean inside. Excuse me, outside. This is the outer layer. So this is the whole, once it starts doing this is the whole part here. All the stuff that's in gray, that's the endoderm. This is the outside, that whole outside layer. So can you think of any prefixes that you've learned that mean outside, outer? Exo is one of them. There's another close one you may or may not know. Sounds like exo. Ecto. Yep. So in this case, they call it the ectoderm, and you're totally correct. The exo is also, but this is the ectoderm, meaning outer layer. Yeah, I should write inner. Doesn't totally matter, but. Okay, so that's the outer layer there. And that we're mostly concerned with limited things. The ectoderm. Let's just know it turns into the skin. And also the central nervous system. We should know that one too. So it's turning to the skin, turning into the central nervous system. If you, CNS is what I'm going to write, all caps. If you don't know what that means, you can write central nervous system, which is, maybe I'll write that for us, means brain and spinal cord, or brain and nerve cords, or ganglia and nerve cords. So then, there's different versions of it depending on the animal, but it's going to turn into that central nervous system, which is like your major nerve and like collections of neurons. So it's going to be things like brain, Um, um, cerebral ganglia, sort of like a brain, but just like not as well developed. Ganglia means collection of neurons. We'll see that in the earthworm. Nerve cord, spinal cord. So all those kind of features, just not like the side nerves and stuff. Okay, and then we come to the inner layer here. So this, I'm going to draw like this. It's actually cells. So, and it can look different depending on the organism, as we're going to see, but this is like a pocket of cells. This is one way it might look. It's on the inside, like in the blastocele. Actually, I want to do it in red. Or magenta. I like my magenta better, I think. That is called the middle, which is the mesoderm or mesoderm. Say it either way. And we're going to know that that turns into the muscles. Other things too, but muscles, mesoderm, start with M. That makes it easy to remember. Actually, turn this into blue. Okay. So those are those different layers forming during differentiation. That's early, and eventually, once it's later, and we will take a break. Promise we will color our stuff. See if we can do a activity as well. I'm not sure. Later, though, a later gastrula, or when it's done with gastrulation, really. We're going to have the gut tube. So let's use our endoderm color for that. So your orange or yellow. So now I'm going to draw a full tube here, a whole cylinder. Then we have the ectoderm, which is not closing the cylinder in, but it's going to be like kind of all the way around it because you have that cylinder. This is a cut through again, pretty much. Not great. Try again. And then you have the mesoderm, which in many cases, in most animals, is going to like line the cavity. It's like right up against the mesoderm. That's better. Okay. And so then this is going to be have a late gastrula, and you have that arc enteron in the middle, and then you have what's called a coelom, which we saw seal. Seal means cavity, but now we have this thing called a coelom that's in here. And that is now, so now you got the gut, essentially you've got the outer layer, then you've got the coelom, and the coelom would be where a lot of the organs would develop. So non-gut organs are all in the coelom. So, you know, whatever it is, liver, reproductive organs, pancreas, we got like seminal vesicles, seminal receptacles, whatever the stuff is that's not the gut, it's all going to be in the coelom, if there is a coelom.
Organelles and stuff.
Organs, yeah, and I'll write that in a second. So it's going to be the body cavity lined by the mesoderm. That means just mesoderms, you know, what's surrounding it. So we're gonna say many non-gut organs. Actually, this will be in the next thing. What are the advantages of this? And you'll need to know this. This is on, I mean, directly on the exam, the advantages of a coelom. And we said it develops the internal organs, so there's more space in there instead of all the tissue being stuck together. More space to develop internal organs. We have organs are more cushioned because of that fluid in there. So if you get, hit on the outside, the organs aren't right up against there. And then number 3, we have the organs can move independently. They're not all stuck to the same block of tissue. So basically they can kind of get more complicated in structure and somewhat function to some degree. So it's like when your gut is contracting because it's in the coelom, you're not, well, really, you're not seeing like, or your heart, when your heart's beating, usually you don't like, whoops, see it doing this, right? I mean, if it's hard, but it's your gut contracting, it's not like rippling your body all the time. If everything was attached together, then everything would move together, which some animals have this acoelomate. So that would put it all together, and then you just see basically all the movements.
At this point in the stage, would the organs be functional, or are they like?
Right now, it would be no organs in there, pretty much, and then they start forming the organs during organogenesis. So this is later. This is a frog embryo. So you've got your ectoderm here. That's the gut tube. Okay, then the actual ectoderm, mesoderm. Right here is the coelom. And eventually that's going to make things, so like here, this is the notochord developing, which is going to turn into eventually like the spine area in the vertebrate groups. We have, again, these pockets of tissue are going to differentiate into organs. I don't have too many organogenesis things in here, but stuff's going to form inside that pouch.
So it's right now they're just cells of the organs.
Right now, yeah, it would just be with the lining and then some stem cells that'll then differentiate and grow into the organs. Yeah, so not organs in it yet, but eventually we call the coelom our inside of our body, like the empty space there. Because if you think about it, you cut yourself open, you're not like all a block of tissue, right? Your stuff is separated and there's a bunch of fluid in there. Same with most animals. We'll see with the earthworm and the crayfish, both are like that, where they're not a block of tissue. You have the space in there, which was the coelom, or is the coelom. All right, I think we need to take a break, and then we'll do the different ones. I think maybe we'll start looking at the animals. Let's do 10 minutes. Let's come back at 9:35. Oh, I don't think we're getting to the animals today. The different groups.
We were supposed to do the problem.
We're gonna dissect
And then we were supposed to.
earthworming.
Yes.
I know. We'll see. It'll be about these things.
Yeah, I that's what I was going over last night.
Yeah, I know. I mean, if we don't do it today, we'll do it next time. Stuff in the back here of most of the different phyla. Look at for the different, sometimes bigger than phyla, but of the different animal groups. And you have obviously specimens that will sort, like I said, on Thursday to see if you can identify. But then I also put a bunch of specimens out here, so feel free whenever break, if we get here early or you want to stay late or whatever, to kind of take a look at the diversity of the different groups there. David, I do have a job for you. Didn't that break it down? Thank you.
And maybe that's really my event is well that answer. Ah, no. No issue. I have to say, being able to stand up in an airplane, it's pretty convenient, that element of it. Sure. Plus minus, what's new? Okay, let's color our little face real quick. Because I want to do that, and that way we'll mostly be done with the different types of development. We have variation in the different types of animals. So here's a phylogeny that has most of the groups. We are not learning all these groups, we're only learning some of the groups. But you can see here, the blue lines, the dark blue means everybody in that group shares the characteristic, like it would be a sedate morphe. And then the light blue means some of the group has that characteristic. And you can see in this case that we have these things here. So we have the through gut and anus. Okay, so that's one thing here that means it's a complete gut. A through gut means mouth and anus. To like has both of those. Let's see. Instead of just the mouth. So most groups have that. So as you can see, if we trace that line down, most of the animal groups, not all of them, but most of the animal groups have this through gut. It's let's see. This line here. So all the blue ones, the dark blue, you can see most of the groups have that. And on your computer, since you can see this, you can zoom in a little bit closer and see that, but that's the through gut. We don't see that in some of the basal animals here, like porifera, which are sponges, cnidaria, which are the stinging ones with tentacles, ctenophores, those are the comb jellies. And then we have this other one here, the mesoderm. Mesoderm is that red layer of tissue we drew. I mean, it's not literally red, but the one that we drew in the embryo that was red. Not all groups have the mesoderm or mesoderm. Groups that do have the mesoderm, that is when we know that it's what's called triploblastic, meaning it has all three layers. So some groups only have 2 layers, endoderm, ectoderm. Most groups have 3 layers, endoderm, ectoderm, mesoderm. So this would be how we know triploblastic. So triplo, tri meaning 3, blast, we said that means embryo stuff. So triploblastic means it has all 3 germ layers, endoderm, ectoderm, and mesoderm. So you can see, if we look at that line, and of course, this is from this is from the paper that I have up by the notes on here that you can, download and look at. It's the Dunn paper, D-U-N-N, Dunn et al. And so you can see most of the groups have the mesoderm. So they'd all be triploblastic. A few groups do not. They just have endoderm, ectoderm, or in porifera, they don't even have the derms. And so all the groups that have the mesoderm that are triploblastic, if you go back and look, you'll see that they all track back to the bilaterians, the bilateria group. And that's the symmetry. So we mentioned the bilateral asymmetrical radial symmetry. So bilateral symmetry are all in the group called bilateria. And those bilateria, you can see it starts here and then everybody, so bilateria starts here and then goes down. So it's everybody except Cnidaria, Placozoa, porifera, and ctenophores or ctenophora. So everybody else is bilateria, and you can see, in fact, that line showing the mesoderm or triploblastic is everybody except for those non-bilateria groups. So bilaterians have both bilateral symmetry, mostly they do, at least at some point in their life. They have bilateral symmetry, and they are triploblastic, meaning have all the germ layers, all 3. Does that mean Cnidaria who have 2 don't form muscles? Because remember, the mesoderm form muscles. No, they do. It just comes from a different tissue, a different germ layer. But they are kind of like wimpy muscles, is how I think of them, versus well-developed muscles. Within the triploblast, the 3 germ layers, we have 2 kind of ways that they can develop. I mean, there's actually other ways too, but these are the big ways we think of. And this would be in bilateria.
You have what's called protostomia or protostome type development, combination really, but protostomia group, deuterostomia group. Deuterostomia group is unit 5. Protostomia plus non-bilateral as this unit. So protostomia, some have protostomy development, some have deuterostomy development. That deuterostomy. DDBT means development. Some have other types of development, whereas deuterostomia, they pretty much all have deuterostome type development. So it's not split completely protostomia, deuterostomia, like the two groups, because protostomia is mixed. It's a really big group, but it's relatively split. And you can see here who does protostomy, who does deuterostomy, the types of groups, and there's other groups that do neither or other, you know. We're just not getting into those specifically. And other groups or certain species. So you can look at this later if you want to. This is what's on the coloring sheet. So we need to know what the differences are between protostomy type development and deuterostomy type development. You'll have to classify those with the groups. And so basically it's a few different things. So how they divide in cleavage, whether those those cells in the ball, the early ball, the 8-cell stage, are stuck in their differentiation, call it determinate, like they can only become certain things, or whether they're indeterminate, meaning they can still become the whole organism. And then we also have how the coelom forms, like the mesoderm in particular, and then whether the first opening, like the blastopore, turns into the mouth and the other side is the anus, or whether the blastopore turns into the anus and the other side is the mouth. So that's what we're coloring here. It's easier than drawing it. You don't need to know all these parts, okay? But let's do some of them. We start, and yours has different names at the top, the correct names. So yours should have platyhelminths, rotifers, nematoda, most mollusks, most annelids, and most arthropods. So we start here with this little zygote, right? A is the zygote. This is protostomy type development. Write that here. So you have the zygote. You don't need to know about the poles at all, anything like that. But then, you know, you divide. So that's going to be our cleavage. You can see it's showing the direction it divides because this is the spindle pulling the chromosomes apart. So we've got division here, first stage of cleavage. Right, so the first stage, you get to the 8-cell, then you get to the 4-cell stage, and that's the first place we're looking at here. So this is the 4-cell stage. And we're really caring about this, just one cell, not this specific cell, but we're thinking about, well, if we look at the cells of the 4-cell stage of this is even before the blastula, this is like morula stages, but if we're looking at those, if we take one out, what part of the organism can that form? So we have 3, we remove 1 cell. What happens to the 3 remaining cells? What happens to the removed cells? So in protostomy type development, you can see this here. I'm going to do this in orange. And I'm just going to do it, you know, quick and dirty. Oops, it's not orange. I'll do it with highlighter, so it's not totally colored. Read my back button. Okay. Got it. All right, so we have this cell. Let's just look at this cell. And then if we took that cell, removed it from the embryo at this stage, and we saw what happened, it would only be able to grow into part of the organism. Now, is that's going to make a living organism? No. But if you separate that cell, it can only grow into part of the adult organism because it's already stuck. It's already limited in its fate. So this is part 1. This is part 1 of the difference in deuterostomy, protostomy. A in the 4-cell stage. B, you can see here, it can only become limited. Cell is limited to certain body parts. So it's differentiated at this stage. And that goes along, sorry, that should be A. Because this is all together. That is what is called determinate development. So that's a cleavage stage thing. Cleavage stage difference is that there's determinate development. It says a quarter of an earthworm larva, but essentially what it means here is that it can only develop parts of the organism because it's already on its direction. It's already like stuck in only becoming certain things, and other cells could become other things. So in a sense, with protostome groups, you can't like take out a cell and then expect the rest of the embryo to recover. It's going to die. Or it could form part of the organism. It's not going to live though eventually. So that's part of the difference between protostomy and deuterostomy. Determinate development, or the second part during cleavage stages is the spiral pattern of division, okay? And I'm going to do this, sorry, I'm just trying to make it consistent. All right, I'll do it quick. So we have a spiral pattern of cleavage of the mitosis. And we can know this if we look at the axis of the spindle. Essentially, the spindle lines up and it's going to line up sideways. And then next round, other way. And then one round this way and one round that way. So it's eventually the cells are divided this way, and then the new cells are dividing that way. And those new cells are dividing this way, and then they're dividing that way. And so it ends up forming this spiral cleavage. So you can see here, you see how this is dividing on an angle? And then that makes the new cells, so it was one cell, right? You saw this cell divides now on an angle and makes two cells crooked, and all the cells will do that. Then the next round, it's going to divide the opposite way. And so then it makes the two cells crooked. And then the next round, each cell divides the opposite way. That's what these arrows are showing. So essentially it builds this ball of cells, and each round, the cells are like in the middle of the cells below them. There's variations on this, but the idea here is that it's spiral cleavage because it goes back and forth and makes an embryo that like has the cells not stacked. This is not spiral cleavage. See how the cells are stacked? Spiral cleavage means this top row is going to be in the middle, and then the next row, and each kind of ball is going to be in the middle of the other ones because it spirals back and forth with the direction. So this is the, let's just remind ourselves, this is that orange cell that we were tracking above. This is the direction of cleavage. The axis just means like the direction of the spindle, which is what pulls the chromosomes, as you know. And you can see it goes back and forth. And that's what makes this spiral cleavage. So it forms this ball of cells, and all the cells are not stacked on top of each other, like in between the other rows. So we can think of that. I'm just going to use this. So these are the division. This is the axes of division. I'm just going to draw those two. That's the axes of division. Back and forth, back and forth. B is the axis of the embryos. I'm not going to color that. I'll just worry about the axis of the cleavage cell. So that's protostomy type development that does that. What you could do here is to do, you can pick whatever colors you want, but I'm going to just do this so you can see what's dividing. I don't know, maybe orange is bad because it all is the orange cell at this point. I'll show for you to see. Okay, this cell is dividing like this. Yeah, this is good. And that cell is dividing, this is a different shade of blue, like this. And then each of those cells now divide on another axis. So now the A cells are dividing like this one cell at the top is now dividing sideways like this. the cell below it, dividing sideways like that, then F, this like cell that was on this bottom right, divides the other way, like, you know, to the left as well. So I'm just going to pick different colors. Doesn't matter what colors particularly. You just want the two cells that come from the other cell to be the same color. Just so I think it makes it easier to see the directionality a little bit. And I can't use the same, like this, the previous cell color, because then you'll have 4 of the same ones. Let's see. And I'm just doing that so it's faster, but does that make it easier to see the back and forth of the division maybe? Okay, then we get to the, after the cleavage stages, what do we get to next? What's the next stage after it goes through all the cleavage? When it ends up like this. But not that. Like this. What's that called? Blastula, yeah, the blastula, great. So that's what we're getting to next. So I'm just gonna do, I don't know, I'll do the same thing again. So we're in the blastula. I'm gonna mark the blastocele now with this color. Just to show us this whole thing is the blastula. Just quickly color it, you know, as usual. Here's our blastocele there. Early blastula. I mean, it's not too late now. It's like a middle stage blastula at that point. It's later blastula. All right. And then what did we say that this, you can see K, no, don't look at K, don't look at the words. All right, you see this little part down here, what they labeled K? That's the part that's like pushing in. So blastula is the thing. What is the word that we call like a hole? Like holes in your skin. What's the word for the hole? I say what? A pore, yeah. So this is a blastula pore, so we call it a blastopore. So I'll just use blue for that. These are not the endoderm, ectoderm, mesoderm. Oh, do I want to do that? Maybe I do. Don't do blue. That's a bad idea. Let's do like not yellow, red, or blue. So what's left? Green? Great. So that's the blastopore. I disagree. I don't think it's the archenteron yet. I think we call that the blastopore at this point. Yeah, that's what I call it. So then we get to the next stage here. So we're going to do the blastopore the same color. There, you know, it's just the kind of opening at the bottom. And then the blastopore in this one is down here. Gastrula here. This is like a later blastula / early gastrula. It's really kind of in between. It's starting to form that blastopore. Late gastrula. Let's see. So if we are doing our colors, what color should I do? The inside here. Let's make it the same color as this inner layer. So I'm worrying about the cavities at this point. What color first of all, what's this inner layer here? I'm gonna do it with the stripes. Okay, I'm gonna use yellow or orange, whatever you were using before. Right, these stripes. That layer there is the endoderm. Now I'm going to do, you use yellow or orange, the same orange, whatever you want to do here. What's this part in here in the middle? That's this inside. So it has that inside word and it's like this sort of shape. So, see, has you see what else do I hear? The blaster port pushes and makes it into it. What's this shape called? Like a rainbow. An arc, an arch. And then we said inside these antero, so we call it the arch enteron, or really arc enteron. That's this inside part here. That's the gut, which is also this here. That's the future gut, which is the arc enteron. That's the part I care about here. So they're calling it the future gut cavity. We're going to call it, to be specific about it, call it the arc enteron. Lined by the endoderm. Okay. And then here in the next step here, let's see, we've got A and B and then C, okay. And we'll see in this next picture what the mesoderm does. So the difference between protostomy and deuterostomy and the mesoderm. But right now, in this picture, it's showing us the mouth and the anus. So, okay, let's think of this word. We know stome already from stomata. Do you remember where stomata were? Excuse me, where the stomata were? In a plant situation? What organ they were on? They're where the water gets evaporated. What organ was that on? Mesophyll. Oh, very specific. Yeah, so it would actually be in the epidermis, but the mesophyll is in between. And what organ is the mesophyll in? Yeah, root, stems, leaves, flowers, fruit. The leaves. And so remember those stomata were those holes, the pores in usually the bottom of the leaf where the water could come out, gas exchange happens. So stoma, stomata, that's all referring to mouth actually, mouth. And proto. Okay, so we have the word prototype, right? How would you define a prototype? New version of something, maybe even a preliminary, a first version of something. So we've got proto, which means like, let's call it first, like a prokaryote kind of thing. Proto, a first version first. And then we said stome or stoma. Is going to be mouth. So this means that the first opening turns into the mouth. So essentially we've got the blastopore becomes the mouth, and the other end becomes the anus. So it does end up having a mouth in the anus, but this is where the name protostomy comes from. Blastopore turns into the mouth. That's letter C. So that's another difference between protostomy and deuterostomy. So in the end, would L2 arrow or would A arrow become the mouth in the protostomy type development? L2 on the left or A, or excuse me, that's not A, that's M. No, M on the right. If we have the first opening being the mouth, the blastopore becomes the mouth. L, do you see how the blastopore is the green thing, right? M would be the future anus, the other end of the gut tube. Of course, J2, the arc enteron, is the future gut tube. In the end, the tube would connect to the other side, and that's how it makes the full tube. All right. Okay. We're gonna have to stop there. We'll do deuterostomy next time. We'll figure out what to do, and the mesoderm and such, and then the different coelom. Because I want to dissect. So We just have two organisms today. We've got annelid, type of organism called an earthworm. Let me just jump over here to show you this situation. I pulled out the annelids from the green box, the boxes you had. So if you can look at your annelid, so look at the containers I pulled out. So everyone shows a few containers, not these. So I pulled out for each some of these. Take a look at them, some bigger ones, and you can turn to like your neighbors, maybe, and the neighbors. And can you see something that's the same, like a similar feature between all the types of groups? You can even like get up and move around with that. We'll just do this like briefly. But I wanted to see if you can figure out something that looks the same and might be a synapomorphy of this group. It's called annelid. Yeah, you might have, it might be best to look around because that way you get different types of versions of different species. So we've got tube worms, we have earthworms, we have leeches, we have bloodworms, what else? We have Christmas worms, we have spaghetti worms. And here's some more. You need to look at more samples. So take a walk around for 1 minute or so. You want to compare them to these as well. I mean, I can look under the microscope if you need to, if there's a small. Rotate around to see if you can figure out something. And here's some other examples. These are polychaete type earthworms. So what kind of feature do we think the annelida? And there's two upper lengths. As well as that. There's two upper. Oh, okay. I'm like, you can switch between the upper and upper lengths, but then there's some people that are small. So we don't have that many people in class. So why don't you just clump together, see if you can come up with 1 or 2 ideas, and then let's see what you come up with. Or you could do 2 separate groups, that's fine. Just go with people now that you thought about it with like a partner or 2, getting a little clump. Come up with an answer of what you think might be the synapomorphy of all the annelida. You will have to talk to each other. I'm just going to set the timer for 1 minute. Let's see. Let's do something. Urgent. Final answer. Let's see. Or if you have more than one idea, that's fine. Okay. Let's get a spokesperson or 2, who however many people worked on it. What are your ideas? Common features, synapomorphy of annelida. What are they? Something in common that you notice?
Segments to their body.
So did everyone see that little stripies?
Yes.
Stripies across their body. So that is true. Now I will say other organisms have segments as well, like us, right? We have different regions of the body, not the like stripe segments. So what's different though between us and like chordates and the annelids, or crustaceans and the annelids, or insects and the annelids? How would you describe them if you looked at them and someone just saw it? You'd be like, ooh, what kind of thing would they say maybe? Like if you see a little thing wriggling around. Maybe a worm body plan. Worm body plan just means like a long skinny thing with a head and like a anus at one end, mouth at the very other end. Is it pretty much the name before our distinguishing feature? Yeah. Worm body plan with segmented body. Yep, this is right. So it's going to have the mouth is on one end, and the anus on the other end. And then you have these segments. Each segment is essentially like, most of them are like repeating units of similar organs, not always. So in the earthworm, when we look at it here, how are you going to tell the mouth? The mouth has this little lippy thing. Okay, it has a little lip. It's you're going to look for that. The anus is literally a circle. It's literally a circle right at the end of the tail. It's going to like a circle. You need to take pictures of all this stuff. We also have this part here. It's going to look like a Band-Aid. To me, it looks like a Band-Aid that wraps around. Maybe we should get the works out. Let me look at it. Yeah, we'll do that. Okay. Then you can look at it while you do it. But we're going to look at that. Most of the organs are up here, like behind, a little bit behind the Band-Aid and the forward. So that's how we're going to dissect it. So what I need you to do is put our hair back. I should do that as well. Roll up sleeves if they're long or might get in things. We need to take any drinks and put those away today. If you don't have, you can't put it in your bag, bring it up to the front. Or I guess I can put a little card somewhere so we don't forget it. I don't know. Let's see. You want to put it on this desk? You think you not forget it there? Let me write this. I don't know. Helps me remember. I have things to say. Or you can put it in your bag. Tuck your bags away or on a chair or something so we don't fall over them. We're going to get glasses in the back and gloves. Gloves, there's two sets, one on the side over here, one in the back. Glasses are in that cabinet. If you have winter glasses, there are bigger ones that kind of go over it. I also have some goggle options if that's more comfortable for you. Let's see. Gloves. Oh, and then we're going to work with partners. If we have an odd number, someone can maybe do a 3, or suppose you can do it yourself, but maybe a 3. Okay. Then what I need you to do is grab some tools. So I guess we need everything here, one of each of the things. We've got fine probes, those are the really pointy ones. We've got blunt probes, those are the ones that are not pointy. We have some scissors, get some smaller scissors, not the huge ones. We have forceps, which are like, tweezers. I would get pins. I get like, I don't know, 20 pins, something like that. Just grab a group of them. There's also scalpels, and then you'll need a tray, obviously. If you want to wear an apron, you can. They're over on the side by the microscopes. If you, it's not messy, but if you're, worried about getting something on you, that's totally fine. And then I need you to bring your tray to the back, and we will, I'll give you an earthworm. Now, these are all hermaphrodites, right? Well, maybe you don't know. Earthworms are hermaphrodites. Not all annelids are hermaphrodites, but earthworms are. Here's a hair type you see. Sure. And so that means I don't have to worry about if I'm giving you male or female. It's all the same, right? So, you're gonna have to come pretty close so I don't drip all over the floor. Last week, actually, we'll probably need them because you want to look at your ELN. So yeah, your partner who's not coming back here to get the worm can pull up the ELN. Just I'll put the thing on overhead as well. The stuff you need to label and take pictures of, but Could be useful. Oh, and the partner who's not doing anything can also come get an earthworm. Let's get a crayfish just to take your time. Earthworm or crayfish, and then one of these binders of the invertebrates. Come on up here. Having trouble with glasses, open glasses, let me know. I have face shields I can get. Something that we're getting foggy. People that are free, but I didn't count. You have how many? Nine. All right, so the third person, whoever's the last person, you can elect to do it in three or to do it by yourself. You are really long. Did everyone get a word? Okay. So what we're gonna do first is inspect it externally, okay? You need someone being the picture taker. If you don't want to cut, just be the picture taker. If you both want to cut, then make sure you trade off. You can, coordinate that yourself. If your hands get like, this is not too gross. You can hold stuff mostly with your forceps, but if your hands get gross, then you can always take your gloves off and get new ones before you use your phone stuff, or take one off and then put it back on, whatever. This is not, oh, by the way, this is not in formaldehyde. It's in like Carosafe, which shouldn't penetrate the skin, so we don't want to get it and eat it or anything like that, but it's not very dangerous, okay? So I don't think these smell at all, so we don't need the no stuff. Yeah. So let me come on, baby. Oh, I just updated my computer. This is different now. Lab archives. Oh, got rid of it. Okay. So we're going to inspect the external structures first. All right, we got to figure out what's the dorsal and ventral side. Do we remember what dorsal side is? How would you describe that in regular words? The back, and then the other one is the ventral, that is the belly. What's the name for the head end? Anterior, and the other end is your posterior. Good. All right, so let's look at this earthworm. Well, you might not want to spell it close up, that's what it is, but it's our. Okay, so we're going to need the following in photographs of the following, nice clear ones, in order to do our labeling for our ELN. So you're going to make the figures, two different figures, internal and external. The dorsal side is going to be darker in color, blends in with the dirt. It's also rounded more. And the ventral side is lighter in color, tends to be flatter. You'll also can look at the clitellum. So that's another feature here. Clitellum is that thing I mentioned, looks sort of like a Band-Aid. That'll also tell you where the ventral is. So the clitellum on the dorsal side is totally wrapped around. On the ventral side, it's just partially wrapped. So it's kind of like not wrapped around entirely. Kind of like when you got a Band-Aid that didn't totally go around your finger. So the ventral side is not totally wrapped around, flatter and lighter. Dorsal side is totally wrapped around, the clitellum, and the whole thing is darker and a little more humpy. Some people's worms might have gotten twisted a little bit, so you might be able to see that and straighten it out. Okay, so first we got to figure that out. We want to take a picture of the whole worm, I'd say. Let's do that for good measure. We need a picture of the clitellum from the dorsal side. We need a picture of the anus, so it's all on the very posterior end. Hold it up. I don't want a picture of the worm and then pointing to the end. I literally want a picture of the whole. And then the mouth is the prostomium. You can tell that too because it has this little lip thing. It's very small, but if you look really close, or we might be able to put it under. I can't see it in this one, but it looks like a little tiny lip, overhanging lip. And actually, there's three back here. And Matthew, do you want a worm and have a partner? Did you want to come up here? Here, come back and get a worm.
So there's a little bit, so take a picture of the prostomia. And magnifying glass. And we're going to dissect it with the dorsal side up. Something like that. Actually, in your binders, there's a really nice picture of the internal structure. It's like in a hard laminated thing. I like that a lot. I'm going to suggest how you dissect it, though, so you're going to read all this stuff. Okay, so I will say most of the organs that we care about is like anterior to the clitellum. So you don't need to do the whole thing because basically everything past here is all just intestine and metanephria, which are kidney organs, and the nerve cords, okay, and blood vessels, but it's all the same. So we're basically going to cut maybe an inch back from the clitellum to the head. You want to cut down the dorsal side. You can use scalpel. You might start with the scalpel and then use scissors. Just you want to be very superficial, which means close to the skin, because if you get in there, you're going to like crunch all the organs. An easy thing maybe is to put a pin in the anus and then cut. But I don't think we should put in not the anus itself, but in the posterior part. I don't think we should put a pin in the anterior yet because the brain is up there. Let me show you. Take a look right. Just scrub on it. This is the brain, the cerebral ganglia. It's two little balls at the very anterior near the mouth, like way up in there. So I think if we put a pin, it's a little like right away, it can poke that and destroy it. So you know, and if you start past the clitellum or more posterior, it's fine if you actually cut in. You'll open up the intestine potentially and there'll be dirt, but that might be a good starting point, and then you can be very superficial. You can also, like I said, do it with your scalpel. If your scalpel is not working well, let me know. I can give you a new blade. So we're cutting down the dorsal side, like about an inch posterior to the clitellum, all the way to the very front, split open at that mouth. So you're splitting it open on the prostomia at the very end. If you want to. Gonna look something like this. Get big. There we go. Okay, something like this. See how it's cut? Now I think this is split a little more than we want it split. Here's the ganglia, they're split here. It might just, this pharynx is cut open. So we'll try to avoid that. But you can see clitellum is like here-ish or so. You might want to cut a little further back, but yeah, maybe an inch past the posterior to the clitellum. Cut or use your scalpel. You might even be able to use your fine probe, the pointy one. Now here's the thing, right? I don't know if you've noticed already, but there's these little like compartment things that are attaching to the side of the wall. They're called septa, or septum is singular. Like septum is that middle part of your nose. Like people get a septum piercing right in the middle part of their nose. This is the same thing. It splits each segment. So because they're holding the segments together, what you're going to have to do is you're going to have to cut along the sides. So you take your scalpel or maybe your fine probe, and in order to separate those septa, you're going to have to cut them. And I usually just do it in little brush strokes in order to open up the worm. Because you see how open this thing is? So in order to do this, here's the septa back here, and you'll just have to like very gently along the side, and then you pull back the sides, and you want to pin it. See how these pins are out, not up and down? They're like at an angle out. Why? So when you take the picture, it's not all obscured by the pins. So it's kind of like, I can come around and look at yours, but you're going to put the pins, you're going to open them up, put the pins like this, open like this. Let me take a look. Let me get my gloves, wear my coat. And you're just trying not to damage the internal organs as best you can. Like I said, they're mostly towards the mouth anterior head end. Anterior to the clitellum for sure. If you see brown stuff, that's dirt, because earthworms eat dirt. Get dead matter, bacteria from that. I call them detritivores. I don't really call them decomposers. I know some people do, but they really don't break stuff down into inorganic compounds.
This is the. So what kind of policy are you?
And if you want to trade off, just you know, tell your partner, hey, I want to be able to. Exactly. And you may want to come in from. I think. Yes. And you can hold this. More sense some people have learned to do if it's being troublesome, we might take a scissor and just come back here. Snip in and then cut down. Okay. Yeah, you might snip in with some scissors, then you'll be able to get it.
No, this
and
is
your scalp,
late.
something like that.
I'll just try not to damage things.
You see how
I
it's
guess
stuck?
I should too. So.
That's because the septa are holding it, so you can take it down side. Those who might be able to, you start back here. A little bit more than this. And that way you can start breaking the septa and pulling it, and like if you damage stuff back there, not. We have all the organs.
Blade, hold on, the blade's not sharp.
And then you know, cut and then pin, and you can pin it as you go. You might not bust over. You pin it as you go, and that'll help keep it open. Sometimes that's easier.
We need a different blade for the scalp.
Different blade. Oh, that's one. Let's throw it out because I can't change that one. Can I? Okay. Right. I have blade change. And by the way, if you get cut, let me know right away. Okay. We'll take care of that. Try not to get cut. Try not to cut towards anyone's fingers. Hold things with forceps instead of fingers potentially. Try that one. If it's still not sharp, that one I can definitely change out. Yeah, and like I said, like if it's not cutting easily and you're having to put a lot of pressure, let's use a new blade.
Now, that's what hurts.
Yes.
Go. Or suffering.
Okay. Yeah, you see brown stuff, that's just the dirt from the gut, it's just the food.
All they did is that they actually cut nearly half of it entirely. They're not. Your fingers, yes. Use the scalpel and fully cut this thing. Just the tip. Nothing else.
And if you pass knives, always pass to the handle, right? You're not passing with the, hold it, like you're passing the handle or put it down first, don't poke it at something. That's looking good. That's dirt, yeah, but that's okay.
Forceps.
Because most of
Tiny first.
it pictures of the septa. It can be, you know, intact or like kind of cut open, but you need pictures of the septa or a septum. I would probably take a picture of like the whole thing, just, and then make a close up. You're gonna need close-ups of the anterior part, right? So once you get it open, septa. The easy things to see are the intestine. So most of it is the intestine. If it's cut open, find a part that's not cut open. It'll just look like a tube. On the top and bottom, or I should say the anterior or the ventral and dorsal intestine. Take a look here for a moment. You'll see this dark thing running down. You'll see it over the intestine. You'll see it over this gut part. This dark thing running down. That's the dorsal blood vessel. You can see it on here. Dorsal blood vessel. These guys have a closed circulatory system, annulus. So you'll see it running down. You'll need a picture of that. And you'll capture a lot of these all together. So intestine, dorsal blood vessel runs on top of it, as well as the rest of the stuff. Septa. How's yours going now? Can you tell a difference with the cutting? And you may want to take pictures with the flash and see how that turns out, because sometimes the flash helps see different things. Sometimes with the flash, I can see other stuff that my like I can't see with my eyes, because it'll bring stuff out. So. And then, so most of actually, that's the things, right? Okay, so all of you,
Now, let's take a picture.
okay, this looks like the ventral to me. You see how the fingers so if I tell them I can see them. It's like more wrapping and then kind of sure. So okay. Mostly the shells up here. Interior. So you don't have to come. Now what I'm going to do though right now is since you do not this it's part of the ethics stuff. I mean this is the intestine. Let me turn on. Oh gosh, both lights. Silly me. That way. Oh, so much of that. Okay. I think we should turn it over because most of the stuff is going to be done. I think this is lip. So you might want to start here and kind of flip in. I just don't have precision. Literally all exactly the same. Once you get past here, we're going to get those. Yeah, we got it. It's basically just the intestine with the dorsal blood vessel, with the ventral nerve cord, and the medial blood vessel. It has all the things, but it's just like the same as what you're going to see posterior to the clitellum. So the thing is like, do I really need to do that? But cut on the other side there. All right, so I'm thinking we need to release the septa a little bit more because it's pretty tight still. So for example, and we're going to try not to poke into these like blob things. Those are all the organs. So I'm going to think I might take mine. Can you see can you see here how they're stuck? So I'm just going to gently those so that I can get that wall really away from it. This is not that sure, so I might use a scalpel. But you see how it's kind of going in between there? And you see here, we want to keep these intact. So we're just going to get right on here and try to release those from the side. And then you'll pin it like that to keep all the blob stuff in the inside as best as you can. And that's far enough because everything's the same. Yeah, all the stuff's up here. Yeah, it's on the side. Yeah, it's just the list. It'll just take less time. You do need to release this more though. So why don't you come in and then on this side particularly, get right between the skin. This is probably okay. Between the skin. So that's a little good. Okay, I'll go over the parts in a minute because most of us will open it. And then everything past that's going to be like this. Now, the question is though, is the gut, oh, it's not, this is the gut here. So it's a little bit, it's not open. It's not open. So, so why don't you release the wall here? And that way you can actually see the gut. See how that one too? Now, and you can see right, it's a little, not it's like beautiful. So I take a picture of this. You can see that just local vertical that dotted line. That's where the left is around. This is good stuff. Let's open it up and release it from the sides up here. Well, from all the bottom things, that's good stuff. That's the organs, the primary organs. You may need to okay, so most of the guts kind of hush open, which is okay. That's why I go around. There are some things.
Go a little further back. Yeah, the water, hold on. Let's see. Hey, go for it. Yeah, well, I think she wants to. Just like this.
Yeah, get some of that dirt out. And not doing it directly on it. Yeah, on the side. So, and stuff it up with our head a little bit.
I would just see it.
Yeah, really did, you look at all the way up to the gas. Right. I'm gonna need to be tiny like that. All right, so we want to start identifying stuff soon. So I'm gonna go over. If yours is full of dirt, you can try to wash it out with one of our squeeze bottles. Angry cartridges. We call them angry cartridges. That's improved. It makes them step a little bit more. How's it back here? We got it on the dorsal side now.
Oh, yeah.
All right.
We need that shows. Should be good.
Okay, let me go over the parts though. So, and usually if you just take, I don't know, a few pictures of this, it'll be sufficient. I would at least have to sex the crayfish today and take pictures of the male and female external features, so we at least have that figured out. Oh,
See,
maybe try this.
those are the ones.
Okay. And some people might not have ganglia because we, slaves get damaged. So we'll find someone who has good ones and we'll all use their pictures. Ooh, better release. Better release. This side looks particularly nice.
That's one. The big, like, longer.
So this is the pharynx, it's a little cut open. I can tell if you see how it's like, you can sort of see those two ends and they look like they're going apart. Maybe that's the esophagus. That's what we want. Don't get out of there. I took it to but we have it on this side, which is nice. So there's tiny little dots inside. You can really see it anymore. So you might have to, maybe that thing. You might have to get somebody else. It's just fast. This is the pharynx. A little bit. Oh, but you can see this.
Those are serious.
Can you separate that out just a little bit more inside? So you can definitely see the. Yeah, this is the crop. Okay, so a lot of this stuff got split in half, which is okay. But you can see this. See how that would be like this. But I think those are all going to go together. But nice pictures of stuff there, though, so we definitely get those.
That's right, I know.
And then we'll see the seminal vesicles we can see.
This one's.
Okay, take a look for a moment so I can show you the parts so that you know what you're looking at. Okay, so got well, you start with the mouth, you got that already, the prostomium. Then we have the pharynx. The pharynx is the first part here. It's kind of flat. This one's cut open. Hopefully yours is not cut open. If it is, then you'll get someone else's pictures to help you. So basically the pharynx is up here. It's a really flat part before these little black things. Okay, it's the anterior to the black stuff. So after that, then you'll have the esophagus essentially, which runs under the black things, so you're not going to see that too much. Then you're going to have these two parts, okay? These are important digestive parts. It doesn't have stomach. It has two parts here. You have a part that's like a balloon. That's the crop. It stores and softens the food. And then you have a part, this part, this is the crop. Then you have a part that's a little bigger behind it and it's very hard. That's the gizzard. It grinds the food up. Okay, so your crop is like, so here's this thing. Crop is this. If you kind of poke on it, this whole thing. If you poke on it, it'll be soft. Don't poke too hard because it's like a balloon, you might poke through. This is the gizzard. And if you poke on that, it's going to be very hard. So the crop is first, anterior, gizzard that grinds is second. Gizzard's usually a little whiter. If yours is cut through or damaged, you can, check around with some people and see, but you can really push it. And I do it with the blunt probe instead of the sharp probe, or the fine probe. So poke on it. Is it hard? That's the gizzard. It's posterior. If it's soft and like a balloon, that's anterior. Anterior, that's into your crop. Then after that is intestine. So all the way down the body is intestine. If you're seeing dirt, it means it's cut open. So just cut a little further down the body to see the intestine. It basically looks like a tube. See if I can. Oh, and you have a nice picture in your binder. So there's a really nice thing in the binder. Guess what? So you'll be able to see those nice things on here. All right, so pharynx. So prostomia, pharynx, esophagus is under here. I don't think I have to label that. And then you have the crop, which is number 8, the gizzard, which is number 9, that's hard and white. You have the intestine after that here, which they're labeling 11, I think. Yeah, 11 is running under that dorsal blood vessel, septa, and then you're going to see these, the dorsal blood vessel, the black thing. There is one on the underside too, the ventral side, but I don't think I have to take a picture of that. Then we're looking at the aortic arches. Aortic arches or hearts, there's 5 of them. You might not be able to see all 5, but try to. They are the little black fingery looking things anterior. So they arch over. They have 2 parts to each. When they're alive, they'll be like 5, but when they're dead and you dissect them, they're these things. So black pair here, here, here. So you see that thing? That's an aortic arch. Has 2 kind of sides to it. Right. There's one, there's one. And so this thing, you can see 5 here. 1, 2, 3, 4, 5. Each one will be in its own septum. So you're looking for the 5 aortic arches that pumps the blood down the dorsal blood vessel. They get gas from the across their skin, not lungs or anything, but the blood is close to the circulatory system. So get your 5 aortic arches, or as many as you can see. Then you have these blobby things. Okay, the big blobs are the seminal vesicles. So seminal is referring to semen, which is referring to, which contains sperm. These blobby things, seminal vesicles. The little baby ones, you'll see tiny baby balls attached to the side usually. Those little white ones are the seminal receptacles. So the seminal vesicles are going to store the sperm. So there's testes in here. You can't see those, they're too small. But there's testes in here. These are testes. They make the sperm. The sperm comes out, gets stored because there's a little duct in the seminal vesicle. When the worm eats another worm, it's going to they have to exchange sperm in the species. So the sperm stored in here will come out some pores in the bottom, and then it will get to the other worm. The other worm, and at the same time, the sperm that's exchanged will come in some pores and will get stored in the seminal receptacles until fertilization, time for fertilization. Then there's ovaries that make eggs. The eggs will come out. They'll drop the sperm back out from the receptacles, fertilize. The clitella makes this gooey mucus. It makes a cocoon around the egg, the fertilized egg, the zygote, which rolls off the body, turns into this hard, structure. The baby worms develop in there, and then they hatch, and you got a lot of baby worms. So big fat one, seminal vesicles that store sperm from the other one, from itself, receptacles which store it from the other one, and then ovaries and testes. Actually, these are all ovaries, and these are testes now that I look. So there's pores in the walls and stuff. Okay, so you'll need to differentiate those. If you don't see receptacles, that could be the case because they can get damaged. Let's see here. What do I see? These are clearly seminal vesicles, these big white blobby things. Here's a seminal receptacle. See, it has this little one, the little ball. You might also see them on the walls. So you're looking for some little ones.
Yeah.
They're very easy to damage though. So if you don't see one, does anyone have really good receptacles that they can clearly see? The little white balls? Yeah. You guys think about good ones? Oh, yeah. Yeah. Oh, these are really good ones. So if you don't have them, you can take a picture here, or you can ask Tammy to post a picture maybe on Miro board, just throw it up there on the diversity one, or you can put it wherever and I'll move it. Oh, yeah, this is so. So you're these are aortic arches, I think. Oh, they're just not black anymore. But you see how they curve over like a finger? That's because it looks like a hair in the hair. I'm not sure where it is. That long? It's just so tiny. I think that's look at this.
I keep multiple.
This is the problem. If you look at this is kind of cuts, I think you start seeing it. Yeah, that one's good. Although you do see.
Yeah. What's bad is that happened like a day after I realized it. I don't remember what I did, but it was in my pocket. I'm like, that's one. I just liked it. So I'm not gonna do this. I'm just gonna. So
Yours, this is yours, it's kind of on the side. You guys, you see that black thing on there, that's. I don't know about the receptacles, they're so tiny, I think those, your aortic arches could be white too. They might be tan or white, so, okay. Oh, yeah, this looks good now. Okay, this stuff's cut open, you'll have to get someone else's there, but great vesicles are big ones. And then seminal receptacles. Maybe that tiny black. You know what? I think it's these tiny. So I think. I don't know, because those are vesicles. At least we have. No, there's C tiny ones. The vesicles are in there. Not sure about the receptacles.
that may be.
Looks like the receptacles are right next to the something right there.
Okay, oh, that's dark, that kind of thing.
Yeah,
Yeah, not that one.
that looks like the receptacle right there.
All right, so your aortic arches are kind of like hiding here. So that's a good aortic arch. That's a good aortic arch. Those black things. That's another one. So I don't see five. I think they're under here, which is no problem. So 1, 2, 3, 2, 3, 4, 5. 1, 2, 3, 4, 5. So you'll see how those double things. That's aortic arch. That's the next thing. This, I like this because it got kind of a little crooked. Okay, these two go. Oh, this is good. So you're gonna probably show us those two things. You see those tiny like roundy things? It's a kind of underneath some tissue, but I think that's sort of looking. Because they're two little ones right at the front. And see, yours was cut like this,
Yeah.
which means you didn't cut straight through it, which is unfortunate. It's nice to have everyone cut a little differently, because then you see different things. So they have pretty good cerebral ganglia at the front because theirs aren't cut in half. Yours might be cut in half. Also, we want to take out part of the intestine and find the ventral nerve cord. It looks like a string, a thread. Looks like a thread down the belly. Okay, it's right. It's not like fat and thread. It's a very thin thread right on the ventral surface. So take out a part of the intestine, and then we got to clean up. And if you have a picture, it'll probably be pretty good. But I don't even have a picture here of that. Well, let me see. Worm. But it looks like a thread, not like a heart, not like a rough thread, not like a rope. tiny, tiny thread. Let's get to lab, let's get to animals, my images.
Is it attached to the wall?
It's gonna be basically attached to that wall.
Yeah.
Let's see.
Don't send me those pictures, right?
Yeah.
I think we also might want to get a picture of.
Okay, here we go. Here we go. We're gonna get it here.
Case.
Dude, come on. I'll just keep it to it. Ah, here. Okay, so this is part of the intestine removed. This thing is very thin cord right on the ventral wall. Very thin cord, and it looks even smaller than this. It looks like a thread.
That looks like a spot. Look at it. A little bit.
Maybe.
Yeah.
Oh, yeah.
What is this?
But isn't that very, I would think that it's the best way. Okay, once you get your pictures, and you can get a bunch of pictures, you'll probably see some, if you realize you can't see something, let's collaborate with those pictures by putting them on the board. Okay, then what we're gonna do is we can throw our worms away, all right? So I'm gonna have us put them in the biohazard in the back. So it's in the orange trash can thing, which smells terrible with clothes, but take it open, put it in there. They've told me that because it's safe and not formaldehyde, all of our paper towels and stuff can go in the trash. We need to clean our trays. So I like to just like put all my stuff in my tray, stick my tray in here, put some water in there, put some soap, right? Our soap here, squirt it in there. Kind of like, be careful with the scalpel. Rinse it around, wash it off with the soap with the paper towel, rinse it off, and then we're gonna put them on here to dry. Scissors are important to clean on the inside and then dry off and then keep open to dry so they don't rust. Be careful with the scalpels, please. Everything dry, over there. You don't have to put the pins in there and push them around, but you don't have to clean it individually. If you have any tissue or whatever, dirt, wipe it out and put it in the trash. We're going to spray down our area with our disinfectant stuff. We have like two, I think. This bottle, that bottle by David over there. No, if you have like really messed up stuff like that, let's go to the sharks container. Hopefully, I don't want to hurt the custodial staff. And I saw someone else. Who else had a really bad pin? So you had a bent pin, we can put it in. Worms go in the trash in the back. The trash with the orange thing. Someone told me you need that in each of us. Doing it. Alright, so put those in there, clean your tools, grease sinks, right? Sink over there, sink over here, sink over there. Two people should be able to be at each. And then what we'll do with our trays is to put them back and forth so they can dry. Don't nest them in the drawer yet. So you put one like this, and then one on top, and then like this, and that way they can dry off.
What did you want to
Yeah, so once you put all your stuff away, and you can bring the binders back here in the earthworm buckets. Then you'll take your stuff off and then you clean it with the spray. So you're gonna spray and then you'll wipe it down. You can put your annelids back in your tray if you have space. I think you should. So we can kind of keep those green trays in the fine. Technically taking your gloves off, you probably learned this in 406, but you like take one off and then you roll the other one over it, so this should show. I mean, we're not using anything crazy dangerous here, but it's good practice. Let me show you. Okay. If you just pop your eyes up here just for 1 minute. So take a look at me just for a minute. Okay, taking gloves off. Hypothetically, you don't touch here because you don't want to touch your wrist. So you're gonna pull here, pull it like that. Then your hand is clean, right? So you can pull here, inside, and then you can throw those away. So all the stuff is on the inside instead of touching your hands. Technical way to take gloves off. These can just go in the trash because this stuff is really non-toxic. And then make sure you wash your hands. If you have sensitive skin, like me, I have some not antibacterial soap up here. It's much soft. I don't know. When I do the soft scrub, my hands like my eczema breaks out. So feel free to use this. And I would put my work, obviously you're not gonna be able to put your crayfish ELN together yet, but I would put my earthworm ELN together as like now, so that you're done with that. It's not too bad. It's 2 figures, 1 interior, 1 exterior. It's just the, you know, this list of stuff. I think we got over it all. You make it nice and neat. And there'll be 2 slides, 1 interior, 1 exterior, figure 2. It can be more than 1 picture on each slide though. And if you need to make more slides, figure 2A, 2B, that's fine. But it's not as extensive as your plan, figures. Trays are gonna go back underneath here. But they're gonna go back and forth because if they nest, then it's like they can't drive. So why don't we start either side like this way, and then the next person over the other. Perfect. All right, when you make your areas clean, I mean, I assume you guys are competent, but you can like let me know. Make sure you wipe it down. And if you come into class on Thursday and you're like, oh, I don't trust whoever sat here, which is probably you, but if it's not someone else, you can always spray and wipe down again. So that's fine. Do wash hands. Okay, you washed hands and all, then we're good to go. I'll see you Thursday. We're gonna do animal stuff, see where we're at. If you haven't read your animal stuff, please make sure you read it so that and watch stuff, watch videos, so we're like prepped for that, because I'm gonna have to do it. We're gonna make a table next time, work together making a table, do our card sorting, finish the coloring. Did somebody else need that picture? I don't know. So look at your stuff. If you end up having something you can't see anything, put up a public piazza post and let's see who has good pictures of it. And then we'll share those. We can either share them on Piazza and upload them there, or if we want to put them on the board, we can share there too. So if you're like, oh, I looked at my pictures, you know, before Thursday, and you're like, I can't see this at all, put up a post and we'll know what we need to target. Great, thank you. Smelly. Oh boy, I think this is smelly. I have big stuff, so people are really affected by that. Everyone's different sensitivity. Yeah. So you take a little sterile Q-tip and put a little on your nose so it's safe to let it smell. Yeah. So if you're sensitive to it, let me know. We can do that. Yeah. That'll work too if you have a little essential oil that's safe for skin. Not all of them are, but you know, absolutely just bring that in. Good idea.
Crazy, because I remember whenever I just thought something, I feel like it smelled really strong, but now I feel like it doesn't smell as bad as me. I smelled some crazy stuff with the bugs.
It's all exposure, right? Are you gonna bring your insects on Thursday?
Yeah, I will. I'll bring my box with the birds too. Yeah.
But even if we do the exam, then we can still look at them later too, because it's very interesting. So, yeah. Or if you want to bring them in too many and you want to bring them in a few days, you can do that.
Yeah.
If it's too many, I can always bring a cart downstairs. We can coordinate. Just let me know.
I was planning on bringing my little box. That I have that a variety. I have a lot of like. I have a few different ones. I have 2 wasps and like 3 honeybees and stuff. Yeah. I just bring a little box. Cool. Some of that and
we can always look at them underneath the scope, stereoscope, or we have those microscope cameras and we have a macro tube that like the clear thing in there. So you can put the camera on there and then macro tube over the specimen. And then I can project it so we can see it close up, which could be cool. In the cabinet, and we're just going back and forth. So we are not nesting them so that water can dry. So this one will just put on top where you can do that one. So.
I think the worst one, it smelled really in the whole water outside. Yeah, it was just we were thinking there wasn't any.
Oh, I was like, is it rotting?
Yeah,
I imagine from the smell about that's good. Exoskeleton.
It just wasn't big enough to do anything because if I'm gonna get it like
scoop it all out.
But I don't feel like the body was big enough for me to do that. So I just kind of was it as I put it in a frame by itself. So
True. And I will say though, that sometimes the invertebrates we do are so much bigger, sometimes they smell significantly, and the crayfish sometimes smells like very secretive. So that can be bothersome to some people. One time we opened the cat though, and it had gone bad, and it was a rancid cat. I really almost threw up. We all ran into the hallway. It was so bad. It was so bad.
Who volunteered to go get rid of it?
This is a good question, probably me, but I think I put the stuff under my nose. I think what am I going to send a student in there?
Yeah.
I don't remember if it was my class or it was a class I was sitting in on, because
I took the bonus points.
No X, one point. Like, honest point, just to get rid of this.
let me put you at risk, not as full risk, but for vomiting. I don't know if that would be.
I think that the worst smell I still get, the worst smell was, when we were, I went to the dog clinic salon. We had a diaper team.
I don't know.
So it's this like trash can that, like, when it's like continuous trash.
Okay.
And you.
Well, like baby diaper things.
Yeah.
Big diaper genie.
But we used to have for the poop.
Yeah.
And for the poop bags. And then when we forgot to clean that thing out, we took that, we played paper rock scissors and then the passenger got off early. Because the smell that came out of there. I have a strong stomach, but even then.
I would imagine too, you have some like, if you get those ear infection, yeast infection or bacterial ear infections in dogs or.
Oh yeah, there was
So
not
that's,
all those dogs are healthy.
and I imagine too in their poop even, you might have.
We did have
parasitic
like,
infections.
and I have tried to tell parents, I'm like, your dog had friends, not external ones.
Do have actually right here, fine thing. I think this is the dog, is a dog. Yeah, this is a dog with parasites in it. I don't think it had all of them, but they put different parasites that can be found in dogs. So there's some nematode roundworm, there's some blue flatworms, I think, there's in the liver. Something fell off, maybe that was in the stomach, I don't know. But then a bunch in the intestines. So we've got tapeworms, which are flat lymph, we've got roundworms and nematodes, ascaris. I don't know what trichuris is. I don't know what that is. They look like a nematode roundworm. They're paper. Yeah. So there you go. Dog parasites in the intestine. Who knew? Is there somebody who's here still? Oh, I got check. Interesting. By the way.
There's gonna be some doing, let's keep my phone clear.
Oh, there's no person here. It's a container back there. Hardwood, which you can fill it up with. I think this one was really low.